Well, welcome back, everybody, to Thrive in Your Body with Homeopathy, where we discuss what homeopathy can do for you and your loved ones, especially if you're suffering from a chronic health condition. So, hello there. My name is Monica, and today I'm very much looking forward to speaking with our distinguished guest, Dr. Iris Bell. So, when Dr. Bell accepted my invitation to this podcast, I was not only deeply honored, but also very excited because her relentless work in elucidating homeopathic principles in light of latest biological and medical science is truly outstanding. And as we face ever deepening crisis in healthcare where chronic illness is not being effectively addressed, this research is very, very needed. So it is my belief that giving homeopathy back its place as a legitimate and indeed cutting-edge medical science has a potential to help solve current health crisis on the planet. That's why I'm very excited and very honored to introduce you to our guest today. Let me just tell you a little bit about her. Dr. Iris Bell, MD and PhD, has been a researcher and teacher in areas related to holistic, complementary, and integrative medicine for over 30 years. She was chosen as one of the best doctors in the Pacific region of the US in 1996 and in the entire US in 1998. She has published over 150 professional papers and dozens of book chapters. Her current area of research is on the nanoparticle nature of homeopathic medicines and the interaction of alternative therapies and treatments with the person as a complex adaptive system. And these are the topics we're going to explain and discuss today in the podcast. So I hope you're looking forward to it. Dr. Bell's award-winning book is Getting Whole, Getting Well, Healing Holistically from Chronic Illness. She has published scores of professional papers and book chapters on her clinical research, in addition to a monograph on environmental illness. On a lighter note, she wrote the funny, touching, inspirational, and award-winning books, Chew on Things, It Helps You Think, Words of Wisdom from a Word Canine, and the Chew on Things Workbook for Fellow Warriors, which was inspired by her beloved dogs. Dr. Bell's passion is to teach people who find themselves with a chronic illness and are at the start of their own difficult journey home to themselves and to better health. A learning experience for us all, indeed. She is especially focused on helping people with type 2 diabetes find a better path for themselves. She herself has type 1 she has been a type 1 diabetic for over 25 years. She does research, teaches, writes, and coaches, and lives in Tucson, Arizona with her 3 rescue dogs. And today, Dr. Bell is going to present for us, and she will talk about complex adaptive systems and nanoparticles, among other topics, and what they have to do with homeopathy, and also how they can elucidate and validate homeopathic principles from the standpoint of modern science. So this is a very exciting and topic that I cannot wait to hear about. Let me welcome Dr. Bell on the show. Welcome, Dr. Bell. It's a true honor and pleasure to have you here. And thank you so much for your time. I know you have a very busy schedule. So welcome. And
Thank you.
Yes, before we get started, I wanted to ask you, can you tell us just very briefly about what you're going to be talking about today and why is that important?
Well, the main issue for many of us interested in homeopathy is how can this possibly work? The skeptics raise that question, and people who even experience the benefits are very curious about what's going on there. And it's really taken modern science over 200 years to catch up with the actual empirical experimental observations that the founder of homeopathy, Samuel Hahnemann, came up with centuries ago. No, there was no science to help him out that would match up with what we now understand is possible. So I'm going to be trying to give a summary of that information, although you're going to see slides that look pretty complicated, and I'm going to try to break them down to explain each slide with its punchline more than going through every inch of the information on a slide. This is all adapted from a more technical talk I often give to other homeopaths professionally, but also to conventional doctors who don't know very much about what this field is all about.
Very well, and thank you so much for coming on the show, because I think that general public, who is the consumer, so to say, of homeopathic medicine, also deserves to know how it works and why. So I really appreciate that you popularize the science and you make it more available to each every one of us who may not be scientists or doctors. Okay, very well. Should we just start with the presentation?
Sure. Let's
Okay.
give it a try here.
Wonderful.
And you can see my slides now, is that right?
Yes.
Okay. Now, the first thing I wanted to mention, this was one of my rescue dogs a number of years ago now. Unfortunately, I lost him a few years ago, but he lived a long, very eventful life. This was one of them. He had surgery for a ruptured disc in his spine. And while the neurosurgeon was doing this, they decided he had a very large lipoma, a large fatty growth on his chest also. And that was the direction the surgeon was going to be going in. So they fixed the spine problem and they removed the lipoma. And then the next day they sent him home to me with this massive amount of bruising. And as you can see, following the instructions of his homeopathic veterinarian, I gave him repeated doses of the remedy arnica and a different remedy called hypericum that's often used for nerve injury. And in just a matter of a few days, you can see that the large amount of bruising was starting to fade dramatically. He recovered completely from that surgery and moved on to other adventures. But I just wanted to show that I've had many personal experiences like that with my own pets and myself, friends, patients, and others. Now, as I mentioned at the beginning, what many people think about homeopathy is it's a lot of complicated things happen to find the remedy, and a lot of complicated things must happen after that. But in the middle, when you suddenly feel better, the fever goes down, you stop vomiting from food poisoning or whatever it is you're experiencing, pretty much people think, well, that just sounds like a miracle. Doesn't make any scientific sense at all. So I'm going to try and translate what I think is how modern science might help us understand all this. Now, this is the summary of the points I'm going to make one after the other as we go through the discussion. Although I'm actually going to start with the person level rather than the remedy itself, the homeopathic medicine. Now, the first stage is manufacturing the remedy, which turns out to be critical for generating what actually turn out to be nanoparticles. Those nanoparticles then get into the body when they are relevant, what we might call salient, to the condition, the state of that person at that moment in time. That then triggers the response of the living system as a whole system. And that's where complex systems thinking comes in. And the whole process of healing actually evolves in a very, it's not only organized, it's actually self-organized way, which is a feature of what our living bodies are all about. We are self-organized, complex adaptive systems. And then we recover. And all of that goes on without our having to understand every inch of this. But that's my overall picture, and I'll end with that at the conclusion. So my overall approach to doing this was to pay a great deal of attention to fundamental homeopathic practice theory, the principles as well as the clinical observations that have been made. and then to keep my eyes open in mainstream scientific research for things that might overlap and help us understand all of this better. And then I also did, although I'll only be hinting at those during this talk, hypothesis-driven experiments to test these models to see how well they all fit and explain what we're talking about. Now, it's only fitting to start back with what Samuel Hahnemann, who was a physician and a chemist, originally observed about how homeopathic remedies might work. He said the initial action is a product of both the medicinal energy and the life force, which I would translate at this point into the living complex system and how it organizes itself. He said it belongs more to the impinging potency of the medicine, and our life force strives to oppose the action of this with its own energy. This back action belongs to our sustentive power of life and is an automatic function of it. It's called what he called the after action or counteraction. I would submit to you that he observed adaptive activity in the human body 2 centuries before other people really talked about it in the same way. Well, how are more modern scientists describing this whole process? If you stress a normal cell that comes out of the body, a living cell that you might have in cell culture or in a living body, a whole organism, if you stress it mildly, it is stressed and it tries to make changes that allow it to be more capable of surviving under that kind of stress. Now, if you give it an extremely overwhelming stress, you kill the cell or you kill the organism. And that's a very undesirable outcome, obviously. Well, what I propose is that homeopathy is one of those milder stressors that tells the body, hey, you better make some adaptations here. The information about disease the program that you're running right now is not good. It's abnormal, and you want to get back in balance in some important way. To do that, you better get some adaptations started, and that will increase the resistance your body has to the stress. And it will also, very importantly, increase your resistance to other stressors that may be similar to that stress, but not identical. So, for example, the disease a person has is a stressor for certain, but it may cross-react with the features, the characteristics of what that remedy material, source material, could cause if given in a higher or toxic dose. Now, as I've mentioned several times now, living systems would be called or are called in modern science, although many conventional doctors don't pay too much attention to this in their day-to-day practice. We are complex adaptive systems, or really networks of biochemistry, of cellular connections, organs, and organisms. And we make our adaptations through the way the body actually works. The downside of being a living system is that as we age or as we acquire disease, these actually reduce the complexity, which is really the adaptability of the system itself. And so the goal in treating with homeopathy is try to restore as much complexity and adaptability as you can at the given age and condition of the individual. So in a schematic form, what this looks like is you have within the body, you have networks of organs that talk to each other. You need them all working in coordination. You have cellular networks that underlie how these organs work. You have molecular networks, which even underlie how the cells work. And at the bottom of the hierarchy, but very fundamental, is how you activate the genes that you have, which are really your potential for expressing whatever you're doing in the world. Now, you put all that together in a living system, you have a human being. Well, that person is part of various social networks. And that can be very small groups, family groups, couples, but it can be even larger communities and different kinds of groups that we all are a part of as part of our everyday life, let alone the larger picture. So we are networks within networks within networks as a single organism, and then we as an individual organism are part of many other larger networks and the larger environment. This is just another restatement of what I've just said, but what's important to understand is in a complex adaptive system, the different parts are somewhat independent. They have their separate roles, but they also have interrelationships and interdependencies. You can't lose your brain or your heart very well without dying or being dysfunctional in the real world. So there are certain parts that probably have more of an importance to our continued survival than others. You could lose a finger or a toe, and it wouldn't be delightful by any means, but you could still live with that. But if you lost one of these major organs, you'd have a much harder time surviving or getting by. Now, how does the system work? It uses cell-to-cell signaling communication, and I'll get into the details of that a little later in the talk. But the biology occurs at what actually is referred to in modern science as the nanoscale level of organization. Now, a nano, I believe, is about a billionth of whatever you're measuring. So if it's a meter, that's a pretty tiny, tiny measure of distance, if you will, or length, or whatever, very tiny. And as I mentioned, the non-lethal stress that doesn't kill you, as Kierkegaard, I believe, said, that which does not kill you makes you stronger. And that actually is the driving principle of this whole talk. There is a phenomenon that has been extensively documented and described in the modern scientific and medical literature called hormesis. And it is a type of adaptive response that occurs in complex adaptive systems that allows for this adaptability, that when something comes up that would otherwise be toxic if it was in a higher dose or a more severe form or extent, that lower dose will stimulate the opposite effects in direction from higher doses. This has been shown with everything from pesticides to chemotherapy agents, which, by the way, will protect cancer cells if given in a too low a dose, with something you really don't want if you're going to deal with conventional chemotherapy, of course. But so things that are toxic, if given in a sufficiently low dose, will stimulate the body to adapt and potentially strengthen itself to resist the effects, at least up to a certain point. This is a little diagram to give you a sense about what that looks like. The idea is the very harmful dose is at the far right of the slide here. It's when you have an overwhelming amount of the stressor. And as you reduce the amount of the stressor, you will reach a point where you actually benefit the organism. And that varies from substance to substance and organism to organism or cell to cell. So it's a very complicated kind of process, but that's what we're dealing with. If you want to look at a somewhat more elaborate diagram of how the complex systems work, all those smaller parts I talked about originally, they interact with each other. They send information actually back to the larger organism level, if you will. So think about it. When you think about, say, your immune system, it sends messages back to your brain with the use of certain biochemical mediators that inform it about what's going on. And it may regulate your body differently. It may cause your heart rate to go up or down, your body temperature to go up or down, things like that. If you, for example, were fighting off an infection. Now, changes in the external environment are going to affect both the interaction of these local parts. You call them local because they're not the whole organism. They're just the heart, the lung, the liver, the kidneys, and so on and so forth. And one of the fascinating properties of an adaptive system is you have something called emergence, where you can get a behavior. I mean, think about a human being. We're an awful lot more complicated than a liver, although a liver is a pretty complicated organ when you learn more about it. We can behave in certain ways in the world and in our larger environment that a liver can't do. If you took a liver out of a body, it would just sit there. It wouldn't be able to communicate very well, and it wouldn't be able to accomplish very much just sitting there as an isolated organ. So there's this constant interaction going on that affects the larger and the smaller level of organization. Well, has this been observed or commented on in a different way in homeopathy? Well, George Vathulkas is a very well-known master homeopath from Greece who wrote a book in the 1980s talking about how the human being is organized. And he really noticed mental, emotional, and physical levels of organization and talked about in many ways how when you're healing properly, you move the disease process out from the deeper parts of the organism towards the skin and down the body. He was not the only one to describe that. He actually built upon what would be called Herring's Law of Cure, another very famous, much earlier homeopath, who described this whole phenomenon that the human body it's not dumb. It sits there aware of where it's been and how it is organized in time as well as in physical arrangement. So, as you're healing from an illness, you may move from, for example, a deeper psychiatric problem like depression out towards, for example, neurological symptoms, maybe more headaches. And then moving down the chain, you might end up with a little more irritability, but eventually you might end up, perhaps for a while, with some liver problems that reflect the movement of the disease process from more important organs like the brain all the way outward and down the body to an important organ, the liver, but then eventually out to the skin where you might develop a skin rash. If conventional doctors give you steroids to suppress that skin rash, it may push the disease process right back into the body. That's a core belief in homeopathy, which often gets shown and demonstrated when people go back and forth between conventional and homeopathic treatment. Well, how do we get all of this going in the first place? Well, let's start with very simple statements. What we're really going to focus on and how we get this going is low doses of nanoparticles. So they're very tiny particles, as I said, a billionth of a meter in diameter, or sometimes a few of them, in very small doses, actually not similar to the amounts in milligrams or grams that you might take in of a nutraceutical or an herbal product or a conventional drug. So again, the small doses tend to trigger hormesis. The small particles are awfully hard to see. And in fact, without very sophisticated modern technology, no one could see them. Hahnemann thought they were there, actually. He just couldn't see them. His kind of microscopes were very crude and primitive. Well, I wanted to just show this slide, not because I'm going to go through every inch of it, but because I wanted to show you that modern science is slowly catching up with what Hahnemann observed. They, in this study, they gave to skin cells, dermal cells, they gave them a tiny, tiny dose of nanoparticles made from a source material called gold, which many of us would love to have a lot of, but this is a very tiny amount. And it's a tenth of a nanomolar, so very tiny concentration. But when they put it in cell culture with these cells, they then documented what happened to the gene expression patterns. Now remember, genes are fundamental. You activate patterns of gene expression, you shut down certain genes, you activate others in order to accomplish certain biological tasks or goals within the living system. And in this case, they found different patterns that occurred and they lasted for a remarkably long term, which is a very surprising kind of situation. It was to these researchers as well. This is a hint that modern science is on the edge of rediscovering how homeopathy actually does what it does. Let's go back to the basics in homeopathy for a moment. Again, how do we make these medicines? Well, we use very old techniques that were originally developed from Hahnemann. As a chemist, he had a mortar and pestle, and he ground up the source materials if they were not otherwise soluble, ground them up in milk sugar, for example, and just kept grinding and grinding and grinding until he got what he felt were very small particles. If he had something like an herbal material, he was able to put that in an alcohol water mixture. And whichever way he got there, he would then take a small portion from either the dry material that had been ground up or the wet material, which had already been made into a tincture, a concentrated tincture of the source material, the herb, the plant, the animal source, and then put it through serial dilution steps in which every step involves putting it into more solvent, which is a mixture of ethanol or alcohol and water. The alcohol is the preservative. I believe he was touted to use whiskey, but that's not how modern manufacturers do that anymore. They use pure ethanol in a mixture with water. And they would take a small amount, put it into a bottle that was mostly solvent, then take that closed vial, if you will, and bang it around. It's called succussions or agitation. And again, people usually ignore that step, but they focus on the serial dilution step and say, well, look, if you do enough of these steps, there's nothing there. Well, that's actually true of what would be called the bulk form of the source material. It's not necessarily true, it turns out, of the nanoparticles that are formed by these kinds of methods. And then, just to add one other complication to what he did, he succussed this material, he agitated the vial. That turns out to be a critical step in making a homeopathic material, as opposed to just diluting materials away. So, as I said, the objection that skeptics and even people who say, well, it's happened, but I don't know why, the main objection is, well, there's nothing there if you just keep diluting. We know in modern science that there is a point called Avogadro's number of molecules where you've diluted the material, the source material, to the point where there's nothing left. And to an extent, that's true. But it doesn't mean that you haven't made nanoparticles or that you haven't transferred nanoparticles from one step to the next, to the next, to the next. So what seems obvious to people staying with conventional classical chemistry and physics is not actually obvious when you get into modern science again. And let me show you some more. When you look at, well, is succussion particularly important for making a biologically active homeopathic medicine? In this study, this research group was looking at cell cultures again. They created an artificial wound, if you will, between the cells that were in the culture. And then they either put in a plant remedy mixture that had been diluted and succussed, or they put in just diluted material, or they put in succussed material, a succussed solvent, where there was none of the source material used in the original in making what they were testing. They compared it with some standard ways of causing healing, where the wound closes up in the cell culture. Certain cells move to actually heal that wound. I'm sure, as I just had the other day, you get a paper cut and eventually, it closes up and heals. That's the action of the cells in your body accomplishing the healing process. It turns out that this particular mixture of remedies is capable of doing it better than just plain diluted material. And even just plain succussed material, which is most interesting, can do it a little bit better than just plain diluted material. Now, how could that be? Well, it turns out that when you dilute materials in a classical way of making a homeopathic medicine, you make it in glassware. What is glassware? It usually has silicon in it, silica, and other related and unrelated contaminants that sometimes show up there. But glassware is silica. And if you look more closely at material that's been succussed over and over, for example, in one vial, the vial gets worn down on the inside from all this succussion. It turns out that you're actually knocking things off the inside wall of that glassware. And you're creating what amounts to a crude silica homeopathic remedy. And that is one remedy that can contribute to healing under certain circumstances. So there you have the effect that is there, not necessarily completely missing at all. Well, let's say you made nanoparticles, and I'll show you some pictures of them from electron microscopy in just a minute. But there is a model from modern research done by a very amazing group in India, which first described finding these nanoparticles with their nanotechnology equipment. And they found that when you succuss materials, not only are you creating more, you're knocking materials off the inner wall, like if anything got on the inner wall, but you're also creating nano bubbles. These are just little bubbles, tiny bubbles at the nanoscale level, which are created and show up and actually surround the nanoparticles and move them in the solution, often towards the top. Well, when you stick in your pipette to move the material to the next dilution step, you're going to grab some nanoparticles and some nanobubbles. Those are going to go into the next step, and then you're going to succuss it. And so anything that is on the inner surface of that next step is going to get knocked into solution as well and interact with what gets moved there. So it becomes a complex system of its own when you're making the remedy. Okay, well, I promised you a picture. Doesn't look like much, but at massive magnification, a 30C potency, which you could buy at any health food store, or 200C potency, which you can still get from many health food stores. Those are nanoparticles that were documented from metal, from material made from gold originally. It was originally triturated or mechanically ground up. Again, it looks very crude because that's how homeopathic remedies are made. Doesn't look like the perfect nanoparticles that modern nanotechnology can generate, but it is a nanoparticle. And what you see on the right side here is that, so actually this is one of several other research groups have also found nanoparticles using electron microscopy and related tools in potencies going remarkably high. We're talking about 1M, 10M, 50M, and CM potencies. In this case, platinum in the right-hand study. And these are tiny nanoparticles, but they are detectable and measurable and even quantifiable all the way through a series of homeopathic potencies to what homeopaths would call very high potencies, and what skeptics would say ridiculously diluted nothings or placebos. But turns out that's not what they are. So when they get that tiny, when these nanoparticles, which by themselves are less than 10 nanometers in diameter, however crude or funny shaped they are, there is an even smaller version, which is really a collection of atoms. And it is at least a 10,000th of the size of a human hair diameter. So you can't really see it with the naked eye, very obviously. One of the ways we might phrase this is that they would be called quantum dots, and they're the tiniest of nanoparticles. Now, classically, in modern nanotechnology, they're made from things that are semiconductor materials. But it turns out that you can also make quantum dots from carbon, which every living organism is made from, and alcohol itself is made from, lactose is made from, and silicon, which is a building block of silica, the remedy, and the glassware. And so you actually are probably making carbon and silicon nanoparticles in addition to the source material nanoparticles that are quantum dot size. Now, why are they called that? It turns out that when you get to such tiny size particles, it confines the movement of the electrons in those atoms and molecules, where when they move, they release photons or quanta of light, if you will, and they acquire quantum mechanical properties. And it turns out that quantum dots often have electrical and optical properties that can be documented. So that's a very important observation based on other things that have been documented about homeopathic remedies. This is just more evidence from different research groups that as you go up in potency, you end up with smaller and smaller quantum dot sized nanoparticles. And their concentrations vary, but the smallest ones end up in the highest potencies. That raises a big question for us to do more research on to try to understand, well, how do higher potency homeopathic medicines, what makes them different from lower potencies? It certainly looks like the size of the nanoparticles in them is different, at least in the distribution of the particles. I want to put all this back in perspective and give you a sense here. I have to look at it here. The human hair comes up here at 100 micrometers. A red blood cell is somewhat smaller than that, and this would be a rat red blood cell. 100 nanometer nanoparticle, okay? proportionately, because this is obviously massively magnified, is this tiny little dot. And remember, I told you a quantum dot is a fraction of that. A quantum dot is frequently less than 10 nanometers. Where do you find things like that? Well, you find them to some extent in virus particles, which are nanoparticles of their own. They're their own unique kind of entity. And if you look at DNA, it's roughly in the double helix that we all have heard about as part of the building blocks of genes. That's around 2 nanometers. That's close to the quantum dot size we talked about. And an atom down here may be around 1/10 of a nanometer. So you can see we're playing down here at the very extreme end of the ruler. And things like cells, which aren't so easy to see with the naked eye without magnification and microscopes, those are massive compared with what a nanoparticle is. How does modern nanotechnology make nanoparticles? Let's see if they've rediscovered what Hahnemann did over 200 years ago. It turns out they have. One of the first ways they developed to make nanoparticles was to mechanically grind things. Now they have fancier mills, which create much more uniform nanoparticles, but that's how they make them originally. They've developed all kinds of other methods, some of which involve chemical agents that are added and make these nanoparticles on the toxic side that you don't really want to deal with. But they also use sonication. Well, that has a lot of overlap with succussions. Sonication is putting sound waves into liquid to try to agitate and mix up things that are in solution. So you have these kinds of methods. More recently, in the past decades or so, they've come up with what they're calling green or biological methods for making nanoparticles. Well, guess what? They use plants to do that. Well, homeopathy has plant remedies. What did they do? They started with herbal extracts and then they serially diluted and succussed them. This is what plants can do. Plants being based, a silica based kind of material, there's not only carbon involved, but there's also silicon. And plants can make nanoparticles. If you put the precursors of silica nanoparticles into solution with a plant extract, it will make silica nanoparticles. And so on and so forth. Sugars will also do this. So here we go. We've basically found they've done different things, but they've also rediscovered what Hahnemann did. Now I don't want to don't want to go into painful detail here. The punchline of this slide is that in more recent years, nanoscientists have discovered that the more you dilute the precursor material, the faster you make quantum dots. So that is an observation that, again, Hahnemann didn't know, but he was taking advantage of. This, again, is another very amplified electron microscopic picture of nanoparticles, excuse me, made from natrium or salt, sea salt, and potassium chloride. And one of the things you want to note here is that there's this whole layer around the nanoparticle. It's probably silica. And it turns out that nanoparticles are so reactive on their tiny surfaces, they do what is called adsorption. And things that are floating around in solution attach to the outer surface of these nanoparticles. It's actually in modern nanoscience called a corona. And there are all kinds of things that'll attach to the outside of these nanoparticles, but one of them is silica. So what if you're banging away at a piece of glassware with your succussions and you're knocking a whole bunch of silica precursors and silicon into solution? The chances are some of that material is absorbing around the surface of the original source nanoparticle. Now, people say, well, there's not enough there. Well, there's not enough there for it to be a conventional drug. I'll give you that. But with other technology available in modern laboratories these days, you can document and quantify that there is a measurable amount in those nanoparticles that have been shown in the electron microscopes. So what are types of nanoparticles? In the natural world, they come from everything like viruses and volcanic ash to biological cells. Turns out all of our cells communicate with other cells using tiny little nanoscale vesicles that they release from their own membranes. And those contain all sorts of interesting information, proteins and nucleic acids and lipids and other materials. And again, natural ones like the ones we make in homeopathy would fall in that category. The unfortunate ones that are more toxic and available in too high a dose are things like traffic exhaust. They produce nanoparticles, but not the good kind because of the dose and the fact that they have no value to triggering the body to heal, at least not in the level that we usually run into them. Unfortunately, modern cosmetics, sunscreens, and antibacterial products often also have these nanoparticles. And on the good side, they are engineered these days for medical diagnosis and treatment. But again, they're often used at more toxic levels. For example, to get them into a cancer cell and then release their contents. For example, a chemotherapy drug, so that they target the cancer cell but not the healthy cells. People are getting more and more sophisticated in this way. So it's not using the principle of hormesis or the principle of homeopathy, but people are trying to find helpful ways of using nanoparticles in medical care. Just to summarize this point, say you had a plant that was in the ground. If you gave a very large amount of a nanoparticle that is enough to be toxic in quantity, the plant will die. But if you give a small amount, you actually will trigger better growth in the plant. And this has again been demonstrated in modern nanoscience. Something that those of us who've thrown some remedies onto plants may also say we've appreciated that can happen. Um, but again, you need to be better matched with the information that particular nanoparticle is bringing to the organism, because it's both a mixture of the amount, but also the nature of the information. Is it relevant to this organism? So, has hormesis been shown with homeopathy? Yes, there are several studies. This was done by the group in India that originally discovered the nanoparticles and documented them for the world to see in homeopathic medicines. And so in this case, they took some of the metal remedies that they had in homeopathic potency and demonstrated this hormesis effect, where it was toxic at a certain amount, but actually stimulatory and beneficial for the cell when given in a smaller quantity. And again, generally speaking, What we know is that nanoparticles will trigger multiple biological effects. If they're at a toxic amount, they cause all sorts of issues. They set signaling pathways off. They cause oxidative stress, what were sometimes called free radicals. They trigger immune reactions. They cause inflammation. They don't do good things if you give too much of a nanoparticle. But think about it, if you give a tiny amount, all these functional areas of how the body works could be doing hormetic or hormesis types of responses and shutting down inflammation, modulating the immune response, cutting back on the stress response pathways, and so on and so forth. So, hormesis is a form of adaptation, as I've said over and over now. This is just another diagram pointing out that at the lower doses, you will see beneficial stimulatory effects. At the higher doses, you will see inhibitory and even toxic effects. And this NOEL is an important cutoff even in modern toxicology. It's the no observed adverse effect level for a given material. And typically, hormesis only occurs if you're below that cutoff level. So if you're giving homeopathy properly, you're not giving a toxic material, even if you're giving what would be a poison or a snake venom or whatever you're giving, because you're giving it below the cutoff for having toxic effects. You're giving it at the beneficial stimulatory adaptive level. And how does healing occur? Well, as I've mentioned in different ways, there are several groups in India doing different kinds of research. This was a group that actually looked at a skin cancer in mice and demonstrated that they could modulate all these different signaling components, the biological mediators that regulate these cancer cell functions. They could affect them with a 30C potency of a remedy relevant to that animal's cancer. And that's a very striking thing. Again, that's how it's happening. It actually is modulating the biology by which the body communicates within itself. In a schematic form, this is what we're talking about. An outside or environmental danger signal shows up, which can be a low dose nanoparticle. And it's going to trigger a reaction in the immune system, the inflammatory pathways in the cells. And it'll kill them if it's too high. It'll stimulate adaptive changes if it's low enough. And what's also important is that I hinted at this before. There are signal properties to homeopathic potencies. Not only are they nanoparticles, but several different research groups have shown that they actually emit electromagnetic signals. They even emit photon signals or optical signals. In other words, they have this ability to communicate within the body that is very subtle and can actually still trigger the adaptive responses. I'm not going to explain the details of this, but again, what would be called danger associated molecular patterns, these DAMPs, this is what we're talking about, how the body communicates within itself molecularly to tell other cells something bad's coming your way, you better get ready. That's basically the information that's being communicated. Now, there can be used as biomarkers in conventional medical research and have been used that way. But they also, again, are targets for what a homeopathic remedy is going to be modulating at this low dose in these nanoparticle forms. It's going to modulate the inflammatory pathways and tend to encourage the inflammation to settle down rather than get stronger. This slide is, again, just a summary of the electromagnetic and optical effects. Now, one thing that conventional scientists often demand is that you replicate a finding in more than one laboratory, because maybe they were extremely lucky, or according to the skeptics, they were frauds. But what if they weren't lucky and they weren't frauds, and other laboratories can show similar effects? Well, that's what's been shown with homeopathic signals. And all sorts of different technologies have been applied. And it's very clear that homeopathic remedies actually have what would be called a field effect. If you're far enough away, you don't get an effect. This was first discovered actually about 10 years ago by a research group who had physically located in plant seedlings the placebo series of seedling packets right next to where the verum actual remedy packets were with their seedlings. There was no actual mixing of solutions. There was just a physical location that was very close between the placebo packets and the verums. And it turned out that they saw a beneficial effect from the homeopathic verums, the real ones, but they also saw an effect in some of the placebo packets, which dropped off with distance. The farther away the placebo packets were from the verums, the less effect they had, until some of the packets that were far away didn't have any effect, and they were truly placebo packets. So this is, again, a very important consideration when you even think about, if you're doing a study, where do you store these things? Where do you store the placebo? It needs to have similarity in the vehicle to what the verum is, but you dare not store them together just in case it actually induces a homeopathic remedy effect. And these kinds of things we know are increasingly being studied in modern medicine, because low frequency, weak electromagnetic fields can affect our immune systems. And there's a lot of controversy about that. But again, our concern in homeopathy is that we want to modulate these things in a very gentle and subtle way. We're not interested in causing damage. We're interested in stimulating healing. Now, very close to the end here, we talked several times about gene expression. And one of the things that people would say, well, if all of this is going on and we are modulating signaling, somewhere along the line, something has to change the expression of the genes that regulate all this stuff. And in fact, there are several studies from groups in different countries and using different technologies again, where they have shown that they can modulate gene expression patterns with different potencies of homeopathically prepared medicines. Most of these are plant types of materials and some animal materials, but it's very striking. I mean, apis is bee, basically, made from actual bees. So it's an animal-derived material. But in any case, when you put it in with the cells, you can demonstrate activation of gene expression patterns that are modulating and underlying the biology of the effects I talked about. And finally, what about individualization? You're going to say, well, that's well and good, but if you give me a remedy that isn't the right one for me, not much happens here. Well, that's true. As I mentioned earlier, these nanoparticles have very reactive surfaces, and things form around them, coatings called coronas. It turns out that when you put a nanoparticle into contact with saliva or blood or any human body fluid, it actually attaches proteins that are circulating in the bloodstream onto the outer surface of the nanoparticle. So let's assume you have this nanoparticle who is carefully chosen to have an ability to speak to that person's illness pattern, their disease symptom pattern. And, well, okay, that's lovely. What if we just get a bunch of proteins stuck on the outside? Well, you don't exactly. What you get is protein patterns that attach to the outside of these nanoparticles that reflect the biological state of that individual person at the moment the blood or the serum or saliva come in contact with the nanoparticle. And so what you're getting is an individualized signal from the nanoparticle coming in and attracting the protein pattern that says, hey, we have this disease process going on. Here's the remedy for it. And then you have is what is potentially a very individualized signal. Again, this is very early information in this whole process. But again, this kind of process of protein coronas forming based on what the state of the person is, that has definitely been replicated. The question is, can we start studying that kind of thing in different remedy types and states, probably starting with acute conditions and then moving on to chronic ones? This was one of our studies where, remember, I mentioned that complexity can change. Well, it turns out that the brain waves can reflect when somebody just sniffs the right remedy, the brain recognizes the signal. And it turns out that the response of the brain will change in the brainwave pattern over time as a person is treated. In this case, it was people with fibromyalgia who were treated over a 6-month period. But when people got placebo, there wasn't much going on there. It didn't cause this kind of shift in the way the brain changed. Now, this was sniffing the remedy controlled for the act of sniffing, just plain solvent. So we made sure that just sniffing didn't do this. It really required the specific individualized remedy to get this kind of brainwave response. So what it comes down to in words is there are nanoparticles in these remedies at all potencies, low and high potency. Homeopathic manufacturing methods make these things in a crude way, just as they do in a more elegant way in modern nanoscience. And these nanoparticles have demonstrable signals that can affect bodily cells and cell signaling, probably by starting an activation cascade that starts with the gene expression patterns. And these are parallel to how the body uses its own biological nanostructures and signaling mediators to communicate from cell to cell, organ to organ, and ultimately the whole organism. And you get all of this started with a low dose of this relevant or salient information coming from the correctly chosen homeopathic medicine. And then the body takes over and does self-organized changes to adapt, because its goal as a living system is to be more fit to survive in the environment in which it finds itself. So let's go back to the diagram. Make the remedies, the nanoparticles signal the body responds with its internal signaling processes, healing evolves, and the person recovers. Sounds much more simple than it really is, but I'm suggesting that if you go through all of this, you will find that you could document all of these elements with modern science. And it's very important that we do so to improve the acceptance of homeopathy and to improve the practice as well. So I'll stop there. I don't know if you have questions or... Oh, I can't hear you. You have to unmute.
Yes, thank you so much. Well, this
Uh,
was fascinating, and thank you so much for going through that. I know there's a lot of information, and of course, you can go deeper into each slide. But one thing that I wanted to ask you about, because on the cross section between homeopathy and actually use of nanoparticles in medical science, it seems that medicine, the way it uses nanoparticles, is much less sophisticated and sort of much, I would say, homeopathy with this over 200 years old tradition of, matching and being really precise or trying to be precise, can actually teach modern medicine a lot. So can you talk a little bit about that?
Well, I agree with you in principle, but modern medicine has an underlying assumption that the medicine is doing most of the work, not the body. And so they, even though nanoparticles are much more powerful in relatively small quantities, not the kind that we use in homeopathy, but smaller than completely toxic doses, modern medicine just uses it as a more powerful kind of conventional drug, basically. And they are trying to find ways to, for example, in cancer, get it taken up just by the cancer cells, but not by the healthy cells. So I don't know whether they'll find this out. I think they will bubble around with it for a while. Oh, sorry. I don't know whether they'll find this out right now. I think they may eventually, because they realize that nanoparticles in higher doses can be very toxic at um, at lower doses than the same material, the same drug. But if you look at the larger literature, you'll even see, excuse me, that people making Chinese herbs and Western herbal agents and even nutraceuticals, they're starting to look at nanoparticles as a way to deliver things that are not well absorbed. And so you'll often see, if you see something that's made as a phytosome form, that's closer to the nanoparticle form of curcumin or, resveratrol or something else. You're going to find all sorts of nutraceuticals and herbs turn out that way. And there are several studies in literature on Chinese herbs and Ayurvedic herbs, discovering nanoparticles even in those, because we would expect that. It's just that they're larger quantities, so you're getting what would be called a drug-like effect from an herbal agent. But remember, one of the features, and I read this in the complex adaptive systems literature originally, herbs are multi-target agents. They go for more areas of the body, just as homeopathic remedies do. They have more beneficial effects in good ways. And the proposal in the original paper I saw on that point, excuse me, proposed that you would actually develop more drugs that had these multi-target capabilities, which means they're going to go back to natural agents, because those naturally have multiple targets in the body to affect. They can cause healing in a more profound way that a very targeted conventional drug will do, because it's going to hit one type of receptor, and it's going to try to suppress that receptor from working, or disrupt one thing. Whereas homeopathy doesn't usually do that. It triggers the whole system to make its adaptive changes. So I don't have a simple answer to that right now, but I think they may bubble into it yet.
Yes, well, I think this is a point of where homeopathy and conventional medicine can actually meet, and start a
Yes.
conversation potentially. Although what
Yes.
you mentioned that a conventional medicine does not looks at healing with drugs, not healing as a response of the organism as a complex system, which is a huge difference.
Yeah.
Another thing, though, that came to my attention is there is a weak link, I would think, in homeopathy, because we do need to match medicine to the specific condition of the patient. And of course, this is an in-depth science, and that's what homeopaths have been training to do for since Hahnemann. And we're always trying to find the right medicine for the patient. But this is very subjective in many ways. And I'm curious, with knowing what we know about the potent effects of homeopathic medicines. Are there ways to start thinking about matching medicine to the state of the condition of the patient that would put us a little bit forward with that?
Well, it's an excellent question. And when you think about it, excuse me, it probably relates to, let me see if I can go back in some of my slides here. Um, this kind of concept, what if we could create blood tests where you have in your repertory a collection of possible symptom patterns, in other words, possible remedies, but you're not quite sure how to choose. Rather than experimenting on the person, perhaps if you took a blood sample and you put it in contact with their blood serum or something like that, perhaps we could begin to see how well the attachment of the proteins from that person's blood happens. And I foresee the possibility of developing an in vitro test. Really, just like in conventional medicine, there's more and more recognition, not always practice, but often recognition, that doing genetic testing can help you avoid drugs that are going to make somebody super sick, as opposed to helping them recover even in a conventional way, and try to find a drug that's more compatible with how their genetic profile works. I think we'll end up being able to do that with homeopathy.
Hmm. Yes, which would change homeopathy very much, right? And the way we
Oh, yeah.
practice it. But I think that that's very important because we are very subjective in many ways. In like, how do we interpret the state of the patient? And also, it changes over time.
Yes.
The way the person is expressing their state of health or disease is different during different times of the day or year, et cetera, et cetera. So there's so many variables in there that I think homeopathy also has to step up, and meet modern science as well.
Yes. And unfortunately, because of the skepticism about the field,
Yeah.
fortunately and unfortunately, so much of the focus has gone into what is the remedy that
Yeah.
very little focus has gone into what's the healing all about.
Yes.
And we needed a lot more research on that.
Yes. Can you tell us, well, just very briefly, what are the areas that you think, apart from this, that you think that the research is going to go if if it keeps on developing in terms of homeopathy meeting modern science?
At the moment, I see more and more overlap, just from what we've been discussing and some of the issues that you raised. Personalized medicine is right now in its very infant stages in conventional medicine. But as they begin to realize that they've just been inappropriately treating so many patients with a drug that's a one size fits all, when it does not fit all, I think those areas of the individualization are going to be one of the first areas where we can begin to talk about that. I don't know exactly how, where, and whatever we're going to get there, but I think people who appreciate different ways of thinking about how the body heals and what the remedies are, they can come together and find some better methods um, with modern science. And many of the leaders in this field scientifically are based right now in India and in Europe, because there's been much more interest in it and use of homeopathy, especially in countries like India, but also in Latin America. I mean, the so-called developed nations are way behind that way, because developed in the sense of the US and uh, probably Canada, although I'm not sure, um, because there just isn't a widespread use of as many alternatives. And at the same time, quite honestly, there has always been consumer pressure to force the field of medicine to become more open to complementary and alternative approaches, which are now called integrative. And that is a fancy term for how conventional medicine still considers itself superior, but at least it's willing to let you do other things that may be helpful to the individual patient. And so I think there's, again, the movement in that direction, but it was from consumers demanding the access and demanding that work get done. That's how it all started to move forward in this country, and in Europe, certainly.
Mm-hmm. Yes, and then that's a great observation. I think this is why, as consumers and as patients, we need to educate ourselves so that we have the bigger impact on what is happening and how, this crisis of healthcare is actually being resolved. Because, as we all know, if somebody is chronically ill, they rarely ever can actually expect a lot from conventional medicine and do turn to other solutions. So this is why, this is wonderful. Thank you so much for everybody who's actually watching or will be watching and listening to this, because we need to educate ourselves. And as Dr. Bell says, we actually have impact, and we can start demand better solutions. So I think this is the way forward.
Indeed.
Wonderful. Well, thank you so much for all your time today. And I know these topics are just extremely deep and far-reaching, so we can only sort of skip on the surface. But I hope that this will entice interest in our viewers. If anybody has questions, please send them over, and we'll try to answer as best as possible. And thank you so much, Dr. Bell. It was a true pleasure to listen to you and learn. And thank you, everybody, for watching. And we'll see you soon. Thank you so much.
Thanks again.
Bye-bye.
Bye-bye.